The sequence is like this, Doctor. I'm going to introduce you and introduce Professor Madia, Dr. Norazah as the moderator. And after that, you start your presentation. And at the end, the moderator will, that's Dr. Norazah. She will handle the Q&A with the help of Mrs. Suhail. And at the end, I will say thank you for you. Okay?
Okay.
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 So,
Prof.
so right now,
Prof, you missed.
do I have to stop sharing
No,
or
no,
not?
No.
No need.
You
Okay.
control the slide.
Okay.
Okay, Prof, you can start
Okay.
now.
Bismillah.
Okay. بسم الله الرحمن الرحيم والصلاة والسلام على سيدنا محمد وعلى آله وصحبه أجمعين. You are all our friends that are welcome for our bioeconomy, food security, and healthy life series from ICA Pagoh, and the series 4 for this year. And I'm very happy today to having our guest speaker, Dr. Hadjadj Naima. She is associate professor at the researchers in the Department of the Food Science in the Faculty of Nature Life Science, University of Blida, Algeria, where she have serves there since 2014. And she had her PhD from agronomy in agronomy from the National High School of Agronomy, ENSA, who is our partner in Algeria. We have collaboration between ICA and ENSA. And he did her work in food technology, human nutrition, and biotechnology. Dr. Naima, she is a research focus on the valorization of the byproducts from food industry, particularly with the wheat germ and the nutrients-rich byproducts. Her work explored the composition and the thermal stability and the extensive study in the biological properties of the essential oil and the dry extract for the medicinal plant. She optimized the vegetable oil extraction methods during different technology, and she have a wide experience in food technology. As for any seminar, it cannot be very nice and successful without having a very good and nice moderators. For today, we have Associate Professor Chemist Dr. Norazah Basar. Associate Professor Norazah, she obtained her PhD in organic synthesis at the University of Technology, Malaysia in 2009. And a part of her work, she did it in a very famous school of the Manchester from UK. And after that, she did a postdoc fellowship at the School of the Pharmacy and the Polymolecular Science in Liverpool, John Morris University, in the field of the phytochemistry. Dr. Norazah is a life member in the Malaysian Natural Products Society and the Malaysian Analytical Science Society and the member of Malaysian Institute of Chemistry. She currently held the position of the Deputy Dean of the Academic and the Students Affairs in the Faculty of Science in UTM, and she is a group leader under the Future Ready Education Program, FREE. And her research is focused on the isolation of the secondary metabolites from the plant, phytochemicals, composition, and essential oil for the aroma plants, green synthesis of the nanoparticles, and using plant extract and the semi-synthesis approach toward the synthesis of bioactive natural products. Dr. Norazah, she is also a fellow of the ICA Pagoh, and I hope you all enjoy this session, inshallah, with the very expert researcher and the very expert moderator in this field. Thank you.
Thank you very much. Thank you very much, Professor. Thank you.
Okay. Thank you, Prof. Hisham, for your kind introduction. Assalamualaikum. Good morning, Dr. Naima. How are you?
Good morning. Fine, alhamdulillah. And you? How is everything?
Yeah, alhamdulillah. So I think it's morning there, Dr. Naima, and you are just starting your day in Algeria. Yeah? So here in Malaysia, we are about 3 p.m. and we are almost done with our work day. Yeah, we are nearly close to our work day. Yeah, okay.
In Algeria, it's exactly the opposite.
Yeah, it's
All
9
the
p.m.
people are sleeping.
Oh, okay, okay.
It's 8 p.m. and all the people is sleeping because it's a vacation of winter. So
Oh, okay. Yeah. Okay.
hmm,
No worries about that. Okay. Assalamualaikum and
good.
good afternoon, everyone. So, Alhamdulillah, we have our session today. So, we are very excited to have Associate Professor Dr. Naima from University of Blida, Algeria, with us today. Yeah, Dr. Naima, even though we are very far apart, technology bring us together on this platform to share knowledge in
Thank
our fields for everyone's benefit. So Dr. Naima is an
Allah.
expert in food science and will be sharing insights on the topic entitled Valorization of Food Industry Byproducts, Effect of Heat Treatment on the Nutritional Quality of Common Wheat Germ. Okay, so
Okay.
Dr.
Thank you. Thank you very much, Dr. Norazah. Thank you very much. And I will thank all the organizer for all effort provided to make this event success. And I express my really gratitude for all the organizer, Dr. Norazah, Professor Hisham, Dr. Norazah, Dr. Norazah, every all the people that all the organizer for their kind invitation. And I'm very, it's an honor for me to meet you again, even if it is online, but it's a big pleasure for me to be with you. And allow me maybe to take a few minutes to introduce myself or present my university. It's possible before the communication.
Yes, sure,
Okay.
Doctor.
Bismillah ar-Rahman ar-Rahim. So I'm Dr. Hadjadj Naima from the University of Blida. I'm working as associate professor and researcher in Department of Food Sciences, Faculty of Natural and Life Sciences at this university from 2014 to present up to now. And for my education, I have obtained my authorization to head a research laboratory in food science department. Sorry, in the Faculty of Natural and Life Sciences in Blida University in 2020. In 2015, I have got my PhD in agronomy in National High School of Agronomy, ENZA. In 2008, my master. In 2003, my engineer from the same school, which is ENZA. And my first diploma was in 1993, which is a higher study diploma in biochemistry from the University of Houari Boumediene, Algiers. For the main field of my interest, there is three of them actually. So the first one is about biological properties of essential oil and dry extract of medicinal plants. We aim to substitute all biochemical, all synthetic preservatives by natural ones, by this extract, miracle extract that contain many bioactive components and bioactive molecules. Second, sorry, is about valorization of byproduct generated by food industries. We are working especially on wheat germ, which will be presented after in this communication. So, as we know, the wheat germ is a bomb of nutrient. We aim to add it for some food to enhance the nutritional quality of these products, of these foods. And the last one is we have worked on oil extracted from seeds or from generally it's extracted from seeds. So we have done the optimization of vegetable oils extraction by mechanical pressing using response surface methodology, where the pressure, the temperature, and thickness of cake were measured and controlled and linked in a mathematic model. For my university, it called also Saad Dahlab University. It's a big militant, mujahid, and shaheed. In Arabic, we say shaheed. So from the revolution of Algeria. So the University of Saad Dahlab is located at 35 kilometers east of Algiers, is a prominent educational institution known for its diverse academic offering and multidisciplinary approach. So the university is structured into four faculties. We have the science faculty, sciences, the faculty of technology, faculty of medicine, faculty of natural life science. Do you hear me, doctor? It's okay?
Yes, very clear, Doctor.
Okay, very good. So in addition, the university is home of four specialized institutes. We have the veterinary sciences, the architecture and urbanism institute, the aeronautic and space institute, sorry, and the applied sciences and technique institute. For the faculty, our faculty, which is the Natural and Life Science faculty, it comprises four departments. We have the Common Core, we have department, the Biotechnology and Agroecology department, the Biology department, and the Food Science department, which I am affiliated to. For the Food Science department, we have a research laboratory. We have actually one laboratory, research laboratory, which is named Laboratory of Food Sciences, Technology and Sustainable Development, with this logo. And the main objective of our laboratory is the primary goal is the research laboratory is to advance the agri-food sector by improving processing methods that meet quality and safety requirements while also addressing environmental consideration. This laboratory is structured of four axes or themes. We have food technology and safety equipe or team. The second team is the team of treatment and valorization in agri-food byproducts. The third one is about sustainable and innovative approaches in agri-food. And the last one is about plant production and seed management. For the food science department offers our diploma, so our department offers the following degrees. We have licenses and 2 masters and 1 PhD. We have food nutrition and pathology, agri-food technology and quality control, and production of cereal and cereal derivatives. For the master, we have only 2, the food processing and quality control, and the nutrition and pathology master. For the PhD, up to now we have just one specialty, which is food processing. So thank you for this first part of my presentation, and allow me to present you a part of our work, which is carried out in the context of valorization of food industry byproduct, entitled Valorization of Food Industry Byproducts, Effect of Heat Treatment on the Nutritional Quality of Common Wheat Germ. So as introduction, we can say that wheat is the most important cereal crop, making it the major staple supplying calories and proteins for human and animal consumption. Its first transformation lead simultaneously to finished product, which are either semolina or flour, and the byproduct of milling, which are sound, remolds, and germs. The wheat germ is the main byproduct removed from the wheat germ endosperm during milling due to its unfavorable baking properties. It comprises almost 3% of the grain and can be isolated through judicious modifications in milling process. Is considered to be the most nutritional part of the wheat kernel as it contains amount of nutrients such as protein of excellent nutritional values similar to animal proteins, unsaturated fatty acids, minerals, vitamin E and B, essential amino acid, fibers, and sterols. So we can see that the wheat the wheat germ is a bomb of nutrients. In Algeria, the wheat germ is designated exclusively for livestock feeds. Unfortunately, it has never used rationally or effectively, at least in our country. So wheat germ faces challenges due to its elevated oil content and enzymatic activity, resulting in a limited shelf life. The question we are always asking, could thermal treatment help preserve this nutrient boost while preventing spoilage and extending shelf life? So our main objective is to extend shelf life of wheat germ in order to put it or to add it for the different foods and supplying the nutritional part of these foods. But we need to deactivate or eliminate or destroy these enzymes responsible of this spoilage. So our work aimed to extend the shelf life of wheat germ using thermal stabilization to deactivate lipase, principal enzymes responsible of spoilage, and determine the impact of the temperature used on its biochemical composition, lipid profile, protein profile, and polyphenols. So for the part material and method, we have tentatively done thermal stabilization. The thermal stabilization aims specifically to deactivate or deactivation of lipid and lipase and lipoxygenase responsible for alteration in wheat germ. It was performed at, we have chosen 5 temperature plus the control. We have worked with 50 degrees Celsius, 60 degrees Celsius, 80 degrees Celsius, 100 degrees Celsius. Oven, we put simply, we put the germ in the oven for 20 minutes and we do all analysis. Plus, of course, the control, the unstabilized wheat germ, which is used as control. The effectiveness of the thermal stabilization is tested by water content and fatty acidity, specifically the fatty acidity, which is generated after the action of lipase. The fatty acids are released in the medium and will cause the spoilage. So the fatty acidity will give us, will trick, if you can see, trick the spoilage of the wheat germ within the time of storage. So the effectiveness of thermal stabilization is tested by water content and fatty acidity. The lipase inactivation was tracked by weekly measurements of fatty acidity during 1 month of storage. Low fatty acidity means less spoilage and therefore longer storage life. It's exactly our goal. For the biochemical composition, we have used the conventional methods. For the fatty acidity is based on the colorimetric method with where the free fatty acid are the fatty acid are dissolved in ethanol 95% and treated in the free fatty acids are treated in the sodium hydroxide. For the water content, it's assessed by dehydration in oven at 130 degrees, the ash content by incineration in the muffle furnace at 900 degrees. The lipid content was extracted by exam with a Soxley apparatus, and the protein are analyzed or quantified by the reference method of GELDAL, which contain mainly 3 steps: mineralization, distillation, and titration. For the profiles of lipids, proteins, and polyphenols, for the lipids, we have done the gas chromatography with saponification of triglyceride, esterification of free fatty acids in boron trifluoride. For the protein, we have used the EFLC, ultra performance fast liquid chromatography, where where we applied an acidic hydrolysis to break down the proteins in the sample into amino acids and then derivatize them with phenyl isothiocyanate and acetonitrile methanol triethylamine solution and read them in UV detector at reversed phase column. And finally, for the polyphenol, we have, of course, prepared the methanolic extract for about 6 hours. The extract are analyzed by the dry extract are analyzed by HPLC with a UV visible detector and a gradient of acetic acid as solvent A and methanol as solvent B. For the results, first of all, we should remember our samples. We have worked on 5 samples. The first one as control is unstabilized wheat germ, USWG. Unstabilized wheat germ is the control. The wheat germ stabilized at 50 degrees, 60 degrees, 80 degrees, and the wheat germ stabilized at 100 degrees. To see the optimal temperature we can use effectively in the stabilization of these germs. So, first of all, for the water content range from 12.51% to 13.86%, and this reduction on water is due to the stabilization of the heat treatment, which aims to partially or totally prevent microbial or enzymatic activity by reducing the water content, which is aligned with the Arshad 2007 principle. We can see also that ANOVA, the statistical analysis, put the wheat germ 80 degrees and the wheat germ stabilized at 100 degrees in the same class on the being grouped together, which means that increasing temperature from 80 to 100 degrees had not effect on water content, reducing the additional 20 degrees unnecessary and leading the loss of energy. So, which means we can use 80 degrees instead of 100 degrees, and we will see after that it is the most effective temperature. For ash content, We can see also that ash content range are vary between 3.15% and 4.4 almost 4%, and the statistical analysis give us a big difference in between these studied germ. For the protein, first of all, we can see that the wheat germ is a source of protein. It varies between 28 and 30%, which is a big amount. This, we can see also that statistical analysis put all the studied germ in the same place, which means that stabilization temperature had not effect on protein content, since the ANOVA test put them in the same class A. For the lipid, we can see that they vary between 8 and 13%. We can say also that statistical analysis demonstrate or show a significant difference between the lipid content of the studied germ. This difference show that the stabilization temperature, first of all, the stabilization of the temperature of stabilization had an effect of the lipid content. And this decrease, we can see that the lipid content decrease as the temperature increases. This may be explained by the vaporization or volatilization of short chain fatty acids. For the fatty acidity or the rate of fatty acidity of wheat germ during 1 month of storage, and use it as method of tracking the spoilage states during the 1 month. So we can see that after 1 month of storage, the wheat germ, the stabilized wheat germ, the control wheat germ, the fatty acidity increase from 0.129% to 0.166%, with an increase of 0.003 or 37%. In the other stabilized wheat germ, the increase of the fatty acidity is lower. For the wheat germ stabilized at 50 degrees and the wheat germ stabilized at 60 degrees, this increase is about 0.025 and 0.023 respectively. While the wheat germ stabilized at 80 degrees Celsius and the wheat germ stabilized at 100 degrees Celsius, this increase is the lowest one, and it's about the 0.0152%. This result demonstrates that the 80 degrees and 100 degrees was the most effective stabilization temperature. And as they are given the same effectiveness, and regarding the energy saving, we can we have selected 80 degrees for the stabilization at 80 degrees for further analysis. For the fatty acid profile conducted by GC, and we have the table, as shown on the table, we can see that unstabilized wheat germ and wheat germ stabilized at 80 degrees demonstrate remarkably similar profiles quantitatively and qualitatively, with seven fatty acids, with 7 different fatty acids. We can also see that for the palmitic acids is the predominant the predominant saturated fatty acid with almost 19% in the two cases, in the two germs. And for the unsaturated fatty acid, which is the most important part, we can see that linoleic acid, which is an essential fatty acids, is the most predominant with 57% in the two germs, which is a good result for us, and followed by the oleic acid at 14 degrees and the linoleic acid at almost 7%. For the profile of amino acids conducted by UFLC, we can see that we have identified 15 amino acids with 8 essential ones, and we can see that there are a minor alteration post-heated treatment or observed post-heat treatment. For the HPLC analysis of phenolic compound, as we can see, we have tentatively identified 14 polyphenols with 12 phenolic acids and 2 flavonoids, which is rutin and camphorol. As we can see, the 3-hydroxy-4-methoxycinnamic acids and the salicylic acids, which is a big, well-known molecule, principe actif in French, which is very, very used in pharmacology as ingredient in medicine. We have these two polyphenol acids are the major present in the two extracts, germ unstabilized with germ stabilized at 80 degrees. On the other hand, we can see that unfortunately the temperature affects and has an impact on the polyphenol profile, since some of them, like caffeine, benzoic acid, ferulic acid, rutin, and camphorol, disappeared after heat treatment. For the conclusion, we can see that the rapid occurrence of rancidity in wheat germ can be effectively prevented by applying a heating process at 80 degrees for 20 minutes. This stabilization technique serves as a practical solution for medium-term preservation and ensures that the biochemical composition of wheat germ remains globally unaffected, enhancing its potential as a valuable nutritional food supplement. Renowned for its exceptional biochemical composition, is a standout nutritional asset. Rich in proteins and essential amino acids, it provides significant dietary value. Its lipid profile, dominated by unsaturated fatty acids, supports cardiovascular health, while its abundance of polyphenols such as salicylic acid, recognized for their powerful antioxidant and analgesic properties, offers potential benefits in reducing oxidative stress in relieving pain. The resourcefulness of wheat germ, characterized by its diverse attributes, suggests its suitability across various domains, thereby creating opportunities for its application in diverse contexts. And thank you for your patience.
Okay, thank you very much, Dr. Naima, for the presentation. You know, like in Malaysia, we eat rice every day, and in Algeria, you eat wheat, yeah, wheat every day. Is it? That is the food take every day in Algeria.
Yeah.
Yeah. Okay. So, Dr. Naima, it's very
Okay.
very good presentation regarding wheat, yeah, wheat germ, and then how you want to make sure that it will prolong, prolong the wheat from the experiment that you did. Okay, maybe can I ask you the first question, because in the chat we have a couple of questions, but very
Okay.
short question from me.
Okay.
When I look at the data, like the protein, the fatty acid, and the polyphenol compounds that are present in the wheat, when we do the heating, um, is it will destroy? I mean,
Yeah.
when we look at the data, yeah, some of the data, especially for the polyphenol compounds, which is, you know, polyphenol is very good as antioxidant compound.
Of
Yeah,
course. Yeah. Sure. Sure.
the wheat, yeah, some of the phytochemicals is destroyed, maybe due to the
Yeah.
compound which is not stable at the high temperature. So is it good? I mean, we want the phytochemicals, we want the polyphenols. So is there any
Okay.
rather we prevent the wheat, Dr. Naima?
Okay. It's always the same. The first impression when I present this kind of presentation is the same question as scientific. We all know that temperature is the enemy of all bioactive molecules, specifically the polyphenols, because polyphenols are very vulnerable and very sensitive to temperature. But regarding to other part of the good the good content of the germ, like lipids of the good quality, rich in unsaturated fatty acid, the proteins, the ash, the mineral part, all these are uh, uh, practically unaffected by the temperature. The only part which is really affected directly is some of polyphenols, but our principle is to destroy the enzyme. We cannot, unfortunately, up to now, our study maybe it's incomplete or need to be completed, or to give less temperature, to use more warm temperature, for example 50 degrees, or but as the result shown, we cannot use warm temperature to destroy the enzymes. So we have not, according to me, up to now we have no big choices. We have to sacrifice some polyphenol, some component like polyphenols, and obtain the other, for example, the source, because the main objective of our work is to supply nutritionally, to take the good thing, the good quality of wheat germ, in order to add it for other foods. Even we cannot preserve totally the polyphenol, because there is the polyphenols not doesn't do not disappear totally. There is some of them which disappear, and some other which are content. So we have no chance to, we have no choices to use, for example, cold temperature, or to preserve all components. We preserve 80%, for example, of components. For us is not enough, but it's quite enough to okay.
Yeah. I agree with you. When I look at the profile, the HPLC profile of the polyphenol, it's still a lot. I mean, it's still a lot in terms of the number of compound that can be detected present even after the heating process. Okay, so Dr. Naima, I have few questions here. So I read question number 1, from Hajar. So she asks, may I know if it is possible to use wheat germ essential oil as a cosmetic ingredient, given its content of phenolics, vitamin E, and antioxidants? Is it wheat content essential oil?
No, unfortunately, the
Yeah.
yield, as we know, the essential oil as a good component, a good ingredient for all cosmetic products because they contain bioactive molecule, interesting bioactive component. But unfortunately, our study, it isn't shown here. We have done the essential oil analysis, but wheat germ, unfortunately, doesn't contain a big yield of essential oil. So we cannot rely on the part of essential oil because it's poor in it has a low yield of essential oil. We have worked just here this in this the current year we have we have tried to compare the dry extract, this one which I just presented to you, and the volatile extract, which is essential oil. But we have we haven't obtained enough yield. It was like 0.0. There is no quantity of essential oil, so we cannot rely on this part of bioactive component.
Yeah, because I think the content of the wheat germ is normally the fatty acid. So the fatty acid
Yeah,
normally is the vegetable oil. That's why
exactly. We have. I just take
it is the
a small
so
small observation. The essential oil for our student,
yeah.
our dear student. I know you know this information, but maybe the student confuse between oil and essential oil. Oil, it's totally, vegetable oils are totally different from essential oil. The essential oil are the main component of essential oil are chemical component like les sesquiterpènes, le
Yeah,
carbacrol,
terpene.
but the main component of the oil are fatty acids. Fatty acids are not volatilized. They are fixed. When we put if we put a drop of oil on a paper, it doesn't disappear. But a drop of essential oil on the paper, it's immediately disappear. It isn't the same thing. Even in yield, of course, the yield of essential oil is always small, but the yield of the oil, vegetable oils, is of the rank of milliliter, big maybe 100, 200 milliliter, but the essential oil are always 1%, 2%, 3, 5%. Okay.
That's true. So basically, from wheat germ, it's contain vegetable. Because as by Dr. Naima, based on your insight, it appears if illusion wheat germ. In terms of processing, could you elaborate on how this method compares to cold processing in terms of effectiveness? So if you can compare hot process and cold process for the wheat germ in terms of effectiveness.
Okay. In terms of effectiveness, and we turn back to the principle of our stabilization. The principle is to prolong and to extend the shelf life of the product. of for this case is the wheat germ. Any method or any operating unit which can give us the result, which is the destroying of the enzyme, it will be welcome. Cold, heat, even whatever the method. But as we all know, the cold processing is better regarding to the conservation and preservation of the molecule, the bioactive molecule. But as we know that cold and, for example, freezing or deep freezing does not destroy the enzymes. It deactivates the enzymes. Maybe after how we can see. Excuse me, because I have to translate in French in my hand, and the defrosting. After defrosting, the enzymes can be reactivated. But if we hate them, we denaturated them. The denaturation is irreversible. But the defrosting can be reversible. So regarding the preservation of this valuable molecule bioactive, it's good to proceed with cold or with, but in reality we cannot be sure that the enzymes are 100% destroyed. So I think I prefer the, up to now, maybe there will be another study to use the temperature, the cold temperature. But up to now, and after 8 years of working on wheat germ, I have worked on minus and 18 degrees. There is 2 years or something. We have, me and my student and me, we have worked with deep frozen. It good. Deep frozen wheat germ. We have put it in fridge for minus 18 degrees. It works. It gives us a good results. But regarding the energy consumption, it consumes a lot of energy. I prefer to put them in oven for 20 minutes in 80 degrees and obtain minor alteration, then use the cold process and consume more energy.
So yes, I think there is so many factors actually that we need to count in, especially the cost effective, yeah, like whether heating or cooling or defrost or defrozen, all involve that you need to consider in terms to preserve the wheat germ. So I think all have pro and cons, that we cannot have like 100%, we cannot get 100%, but it's always plus minus whatever method that you choose to preserve the wheat germ. Okay, so another question, we move to another question from Kasih Putri Alia. Okay, Dr. Naima, based on your discovery regarding wheat germ, what are your plans to enhance its value in your country?
Can you please repeat the question?
Okay, what are your plans to enhance its value in your country? I mean, germ, on your discovery related to the heat process on the preservation of the wheat germ. So what is your plan to enhance the
what? Sorry, to enhance, but there is because there is internet issues. To enhance what? Sorry, because it's so distracted. To enhance? What is my plan to enhance?
Enhance the value of wheat germ in your country.
Okay. For my country, I plan. We have our team plan to gather all, to exploit all the germ from the unit of cereal transformation, and really use this stabilization temperature because it isn't difficult to do it. It just like a cake. You make a cake or bread in the oven for 20 minutes, and you have your stabilized. We plan to recuperate, to have and to gather all this germ and put it in a manufactory where we can stabilize it in oven with semi-pilot or pilot scale, and sell it for other manufacturers of good of food industries, in order to put it directly, for example, in the youth, for children, for in some cakes, small cakes, for example, for breakfast, for cereal breakfast. We can add it in cereal breakfast. We can add it in cookies, or like biscuit. We can add it in soup, like to enhance the quality, natural quality. It can used easily, but we need to stabilize them. We need to heat treat it by this a simple and modest and easy method. After ovening, I think it isn't difficult to do it, and start to trade it in like raw material or ingredient, like an ingredient for other to prepare other products. But because we must see the 30% of protein. There is no product, there is no foods, there is rare food which contain 30% of proteins, and in these proteins we have the 8 essential amino acids at the same time. For me, it's a bomb of nutrient because it's cheaper. It's a byproduct. It costs nothing. It's a byproduct which is actually used for feed for animals. So we can use it. We can use this protein, this amino acid, this fatty acids, which are very good for the cardiovascular is a disease. We can use this part of this good nutritional quality, and add it at every product.
Okay. Dr. Naima, thank you for your answer. Maybe I have one last question if there is no question from the audience. Okay, back to the facts or the statement just now regarding the essential acid present in the wheat germ. So when I look at your data, quite a big for the essential amino acid. Yeah, it's quite big drop from the USWG to a treatment of 80 degrees C. In your opinion, in terms of the essential amino acid or the polyphenolic compounds, which one is the one that you want to control more in your study? I know three of this is very important: the fatty acid, the protein, and also the polyphenol. But to control everything, sometimes we cannot have one methods that we can get everything in order. So in terms of these three, what you really want to control most? The fatty acid, or the amino acid and the polyphenol compounds?
It's a kind, Dr. Norazah, it's a kind of question that you give for a mom, and you tell him which is the best, who is the best children of you, which is the best children, or this or this. For me, as a nutritionist, as the food industry specialist, I prefer three of them. I prefer to preserve. I aim to preserve three of them because three of them are really, really important for the body. If we say if we talk about amino acids, it's good for health. If you talk about fatty acid, essential fatty acid, it's really good. Especially if you talk about antioxidant, which is the big debate, international debate, actual debate. All the people talk about antioxidant, natural preservative. But if I have to choose, I will choose the protein and the fatty acids because the results are sure and are quite evident to supply and to enhance the nutritional part of foods, and we can rely on another type of foods which can be a source of these valuable antioxidant molecules.
Okay, Dr. Naima, there is one question here. Have you ever considered the or cell alive system to preserve the original quality of the germ? CAS, cell alive system.
The date of preemption. What do you mean by CAF? What do you mean by CLF?
CAS, cell alive system. Cell alive system. I think it's like a freezer, is it? That freeze. This question from Johor. Yeah. It's like a it's like a freezing system. Cell alive system. Cells alive system.
Actually, I'm not. Maybe in French it does mean another thing. If you talk about the date of the preemption, which means for example, we give for this food, we have to eat it, to consume it before, for example, 24 January, for example. The date of preemption, you talk about this, or because I don't really sorry, but I don't understand the question, the last question.
Yeah. The question asks if you cells a system, and I just go through what is cell life system. A life system. It's like it's a freezing system. I mean, because in your study you are using heating. Yeah, I think it's quite similar with the answer with the question before this, but this is specific to the CAS, cell alive system.
A self-alive system.
Cell. Yeah, it's a cell system.
Okay, if the question is about using cold temperature, freezing, we have already work on freezing temperature. It gives us maybe the best results of
as you mentioned now.
Freezing gives us a big freezing temperature, 18 minus and 18 degrees, giving us the best in the study not shown here, best quality preservation of all biochemical composition. But regarding to temperature, but because freezing consumes a lot of energy, so for me it isn't the best method to preserve the weight germ. For me the heat is better, even it give us some effect on, for example, polyphenol or or for example, disappearance of biomolecule, but it's still, for me, it's still the best one regarding its simplicity and easy to use this.
Yeah. Yeah, I think another reason is if the method is simple, that everyone can do it. You know, like it's just that we need
Yeah,
scientific data to prove that all the nutrition in the wheat germ is still there, even though
yeah,
undergoes a few steps to preserve the wheat germ. I think like
exactly.
yeah, from the experiment or from the lab experiment, that we can transform the knowledge to the community or to everyone. So how to preserve the wheat germ using the most simplest and very cost effective ways. I think
Yeah.
that is the best solution for all. Okay.
I totally agree. I totally agree.
Yeah. Okay, Doctor, I think there is no other question from the audience. So on the behalf of the community of ICA and everyone that are listening to your talk today, I would like to thank you for such informative and engaging presentation. We truly appreciate the insight that you share with us today, especially on the food industry byproduct effect of heat treatment on the nutritional quality of common wheat germ. Because as I said just now, in Malaysia we eat rice every day. So maybe the way how we preserve or how the experiment that we do here is different with the one that you face there and the challenging that you face there. So I would like also to thank everyone for your participation and your questions. Your engagement has made this session even more meaningful. So before we conclude, let's take a moment to acknowledge the effort and expertise that Dr. Naima has brought to this session. Okay, so please join
Thank
me.
you.
Yeah, and with that,
Thank you very much.
yeah. So with that, we officially conclude today's session. Thank you everyone for joining, and we hope to see you again on our future events. Have a great day, everyone. I send back
Inshallah.
to the Prof. Hisham or maybe Suhail.
Yeah, assalamu alaikum. Thank you very much, Dr. Norazah. And thank you, Dr. Naima. It was a great session with a nice dialogue between two experts.
Thank you.
Actually, it's your presentation and the moderator, and we make it very successful one. This will be the last seminar of the year, our last webinar. And I would like to thank again everyone who joining us today. And I would like to say Happy New Year. And also
Thank you.
for this year, inshallah, the 1st of January is the first day of Rajab. And we congratulate everyone, inshallah, for the first day of Rajab. And inshallah, looking to see you again in Malaysia in person. And we're having more collaboration, inshallah, with Algeria, Blida University, INSA, and all our partners there.
Inshallah, inshallah. It will be a great experience, inshallah, inshallah.
Inshallah. Thank you again. And inshallah, 2025, we're going to see you again, inshallah.
Inshallah, inshallah. It was a pleasure for me. Thank you very much. Thank you, everyone. And I hope, I really hope you good and happy New Year's 2025, inshallah. Full of success and full of, hi, Noris. How are you? Thank you very much for your kind invitation, Norazah, and the Professor Hisham, and Professor Inshallah Dr. Norazah. It's really an honor to be with you today, and I hope meeting you another time and another time. Thank you very much. Pleasure. Assalamu alaikum.
Alaikum
Walaikum salam. Happy New Year.
So I can leave? I can leave?
Yeah.
Thank you.